Dr. Ben Perlman, who's at CSU Long Beach. And I'll give you a little bit of an introduction, then we'll let him give his talk. So Ben did his bachelor's degree at UC Santa Cruz. So he spent quite a bit of time in the Monterey Bay area. I was looking at his CV, and in that time he did a little work with Mark Carr, where he was doing some work studying coral reef fish. I'm assuming maybe that was that when Mark was doing work in the Bahamas, or was that like
So Mark was my undergrad advisor doing kelp forest fishy stuff, but then I spent time in Moorea with Pete Ramundi and Giacomo Bernardi. That was the coral reef fish stuff.
So like, how do you go back to cold water after that, right?
Yeah.
And then, so Ben's an alumni here of Moss Landing Marine Labs. He did his master's. He did that with Dr. Laura Ferry, who was here, and she was a research faculty at the time and studies functional morphology of fishes, and Greg Kaye. And Ben's work was, his master's thesis was studying the swimming performance and the biomechanics of surf perch, right? And they're kind of unique because they swim by, flapping their pectoral fins, kind of the way birds do. We kind of call it flapping flight. And so Ben was kind of studying those processes. That'll come back a little bit later. He then went and did his PhD. He did his PhD at Wake Forest University, and there he was studying the biomechanics of the mangrove killifish. And they're a fish that can spend some time out of water, and they can actually use their tail to jump in the air and sort of propel them along. And I was curious about the fish and looked it up, and the first thing I found was this amazing video that Ben put together that was showing how the fish jump and jump in the air and use their tail to do that. And so his PhD work was studying kind of how they do that, as well as kind of the older they get and the bigger they get, the better they get at doing that sort of process. After he did his PhD, then he had two different postdoctoral positions. One was at Stanford, where he was studying bird wing morphology and how it affects their flight. And then he did another one examining biomechanics of frog jumping and that was at UC Irvine. And he also did some work with dragonfly larvae as well. So he's worked with a whole diversity of different organisms, right? There seem to be lots of different types of species, but some common themes, right? Movement by flapping your fins or wings in either fish and birds, right? Mechanics of jumping in the species either that have legs, the frogs, or those that don't, these fish. Right, and so after those positions, Ben, since then he's been a full-time lecturer at CSU Long Beach. Right, he runs the STAB lab, which you can see up there, which is probably the best name for a lab I've ever heard. Right, so
Thank you.
stingray and butterfly biomechanics, and one of those can stab you. Luckily, the other ones can't. Right? His lab studies the kinematics, kinetics, and morphology of animals, focusing on the round stingray. That's the talk we'll hear today. He uses things like high-speed cameras, materials testers, 3D scanners, and X-ray imaging to describe their form and function. And he also teaches a lot of different classes in evolution and diversity, ecology, human anatomy, ichthyology, science communication, and field studies at Catalina Island. So with that, I'll hand it over to Ben, and his talk is Putting Science Behind the Stingray Shuffle and Other Observations with the Round Stingray, Urobatis halleri.
All right. Well, thank you, Scott. I appreciate that wonderful introduction. It's very nice to be back at Moss Landing, at least virtually. It's been a while, 15 years since I graduated. So there are jobs for all of you in the audience who are ready to finish your master's. So that's always good to know. And thank you to Dr. Amanda Kahn for the invitation. We actually did our master's together at Moss. So yeah, I look forward to telling you about some of the current work we're doing in my lab for the past couple of years. So to just go ahead and start right off, let's see if I can get this going. So here you are looking at a very different beach compared to what you would find in Monterey Bay right now, since you have stormy weather. So this is a photo from the summertime here in Seal Beach, which is just about 2 miles from my campus at Cal State Long Beach. So on any typical summer day, you've got these beautiful, wonderful waves, and you'll notice that there's a lot of people wading in that nearshore habitat. So they're swimming, surfing, wading, stand-up paddleboarding, doing something. Now, in California, you're all pretty much aware of the white shark, and many people tend to be afraid of this, the white shark. But if you know much about Southern California beaches and waters, it's the juvenile white sharks that tend to frequent our area in nearshore habitats, but they're still feeding exclusively on fish, not people. So it's not necessarily being concerned with a white shark, it's something else. And that is going to be the focus of my talk, the round stingray, Urobatis halleri. So here's a photo of one of these rays, which are beautifully camouflaged in this nearshore sandy bottom habitat. 2 seconds later, you can unbury them. And you'll notice, again, their color patterns are very, very good at camouflage. And again, if you're wading in the water in these shallow areas, you will come across many of these throughout your fall, summertime seasons. What you need to be aware of is this. So toward the back of their tail, not at the very tip, but toward the back, they have a barb, which is basically this needle-like structure from a modified scale. It's a type of placoid scale that you find across other sharks and elasmobranchs. And I want to tell you a little bit about what this particular barb can do. So here in Southern California, you might hear this colloquial term, do the stingray shuffle. So we're literally putting science to this statement. So does the stingray shuffle actually keep you safe? And if you're unfamiliar with this term or around stingray in general, by the end of this talk, hopefully we'll answer all of those questions. So going back to the barb, in terms of their morphological features, they're basically a needle-like structure that's anywhere from 2 to 4 centimeters or even inches in total length. They have a very sharp tip at the very end, and you'll notice on their lateral margins, if you look at the photo on the bottom left, they have these serrations on the left and right sides. And those are very, very good at tearing through tissue. So if you look at the photograph on the top left, you'll notice the barb that's sticking up vertically from the rest of the tail. So when these particular stingrays strike any particular fish or possibly a human, the tip of their tail has all of this skin that's very flaccid, very loose. So when they use their muscles to generate a strike, the barb becomes exposed, exposing this needle-like structure. So not only can they impale you, which generates a puncture wound, insult to injury, is that they also have these glandular-like cells at the base of the barb on the tail that generate venom. And venom can travel along this integumentary sheath. It's basically epithelial tissue covering the barb, and that can be injected into the puncture wound. And I speak from experience. Here's a photograph of me from a year and a half ago. I look all happy and smiley, but it was all nervous laughter. When I was handling one of the rays, I actually got punctured in an artery in my wrist, blood squirting everywhere. It was super gruesome, perfect for Halloween. And took about 5 seconds to stop the blood, but it took about 2 hours before the pain went away. So what I call my stingray tote bag is filled with burning hot water, basically near boiling. So it'll help denature the proteins in the toxins that are causing this pain. So on a scale of 1 to 10, 10 being really, really painful, it was about a 7.5 or 8. So that toxin, again, travels along the length of the barb, and it will be injected into some puncture wound in a person or a fish or some predatory specimen. So these particular barbs, they're solid. So the toxin is literally traveling along the perimeter of this particular structure. So that's something we're going to be exploring a little bit later on in my lab. But nevertheless, here's this barb. So lots of ways to get injured from this particular animal. So just a little bit more about how many people get injured by these stingrays compared to, let's say, the great white shark. So between 1950 and 2001, so a 50-year period, half a century, there were about 200 documented cases related to different species of sharks along the California coast. To put that in perspective, every single year at Seal Beach alone, again, just about 2 miles from my campus, there's about 400 plus injuries due to round stingrays at that one beach. So these are very, very abundant animals and much more common in terms of causing some type of injury at the beach. So if you exclude drowning, this is basically the #1 injury that you would expect to find in a Southern California beach in the spring, summer, and fall seasons. So if you look at the photograph on the top, again, there is the barb toward the back of the tail. And if you look at the photograph on the bottom, here's an aggregation of some of these particular round rays at Seal Beach. So again, we're putting science to the stingray shuffle. If you shuffle your feet, will you be able to stay safe and prevent some type of injury to your foot, to your ankle, your lower leg, or some other body part. So just to show this in another way, here's a former grad student in the shark lab with my colleague, Dr. Chris Lowe. This is Zach Merson, and he was helping us on some of the beach seines to collect these animals. He took a step and he was barefoot and he stepped on a ray. He took his second step with his other foot and just so happened to step on another ray. So he had to literally be dragged out of the water with a lifeguard and some others. And you can see the photograph on the right. He has these tote bags filled with burning hot water, again, to denature the toxins from the sting. So not fun. So nevertheless, he gets his fancy stingray tote bags. And if you should ever come across a stingray where you get stung at the beach in Southern California, hopefully a lifeguard is on duty so you can get your very own bag. I don't think it's worth it, but you do have a souvenir at the end of the day on top of the puncture wound. So this all leads to the goals of our study. The first being, we'd like to evaluate the kinematics or the locomotion of these tail strike behaviors. So if there's hundreds and hundreds of injuries from one beach alone in Southern California, if you extrapolate that from San Diego to Santa Barbara, there might be anywhere from 2 to 10,000 round stingray injuries in any given year. So a lot of this information just goes unreported because maybe a lifeguard's not on duty. It's really hard to connect all these different counties to make sense of that. So a colleague of mine is working on that. But nevertheless, we're just trying to figure out what's responsible for this behavior. What does it even look like? You would think we would have known exactly how the tail strike occurs if all these people are getting injured. So that's the main study. We also want to describe how the stingrays respond when being stepped on different regions of the body. So again, this comes in context with the stingray shuffle. Does it matter where you step on this animal, and does that influence the behavioral response? And then next, do different foot sizes affect stingray strike response? So if you look across your seminar room, all of you are different sizes, shapes, and all of the things. So think about that. Does it matter if you have a small foot, a large foot, or something in between? Does the surface area that's in contact with this animal affect the type of response you get when you make contact with the round stingray's body? And then last but not least, since half my lab and myself, we're all surfers, we all play in the water all the time, is there some type of surf booty material that can reduce the stingray inflicted injuries? So this is what I'm going to talk about for the rest of today. And then I'll talk about very briefly at the end, some other things we're working on and what we plan to do in the future. So I'd like to give you a little bit more background about this stingray, since they actually made some headlines a year ago for some very interesting reasons. Okay, so I know not all of you are in ichthyology with Scott Hamilton, but you should all take that class. It's amazing. But for those of you not true fishophiles, here is a phylogeny of all the vertebrates. So on the top left, you've got the jawless fishes, the agnathans. You've got the chondrichthyans, which are your cartilaginous fishes. The sarcopterygians, those are your lobe-finned fishes. Here's the coelacanth representing that group. And then the actinopterygians, which are your ray-finned fishes, which make up the majority of the diversity of fish in freshwater, marine systems, etc. So I'm going to focus on the cartilaginous fishes, the chondrichthyes. So within the chondrichthyes, those are your sharks, skates, rays, and chimeras. So there's about 1,200 species of chondrichthyan fishes, cartilaginous fishes. Of the 32,000 described fishes, this makes up about 4% of all the fish on the planet. So here we're just looking at the chondrichthyan group, the cartilaginous fishes, and those are subdivided even further. So your holocephalans, those are your chimeras, the rat fishes. Those are about 4% of all cartilaginous fish. We've got the elasmobranchs, which are your sharks, skates, and rays. So here at Cal State Long Beach, our mascot were the sharks. So we've got a nice little shark representative here. And then I want to focus mostly on the batoids. So the batoids are basically your skates and your rays. And there's about 650 described batoid species. And within that batoid group, which we'll focus on here, those are basically across 4 different orders. And I'm going to focus on the, sorry about that, the myliobatiforms. So the myliobatiforms, those include manta rays, all of the stingrays, and a few other rays. So not all myliobatids have a sting, have a barb, but all the stingrays are within the order of the myliobatiforms. You might be familiar with the rajaforms. Those are all of your skates. So Dr. Dave Ebert at Moss Landing, he of course studies skates and lots of other things, but mostly skates to my knowledge. So here's a tree from 2004. And just a few years later, here's this new tree with this crazy polytomy. It's totally unresolved. But the point I show this is job security for all of you. So we're constantly gathering new information to figure out who's closely or distantly related to something, in this case, batoids. So hopefully we'll be able to further resolve this in the next number of years. But nevertheless, we're focusing on the myliobatids. So here is a fish that was caught just a couple of years ago. This is a freshwater stingray. This is the largest known freshwater fish, which just happens to be a freshwater stingray. So this was caught in the Mekong River Delta in Cambodia just a couple of years ago, 13 feet from the tip of the snout, its nose, all the way to the tip of the tail. That's a pretty large fish. And there's actually one caught just the year before, which was about half a foot smaller and maybe 3 pounds less. But nevertheless, you've got these really large fishes, and this happens to be a freshwater stingray. Here's another stingray. This is a photo that I took at the Aquarium of the Pacific here in Long Beach. This is also a myliobatid. It's an eagle ray. You might recognize this from Finding Nemo. And I want to bring to your attention what you see here in this red circle. So notice there are four different barbs that are stacked on top of each other. So these particular species actually shed their barbs naturally, but they don't always shed them. So in this case, you've got 4 that are all stacked on top of each other, but notice that it's attached to the base of the body. Whereas for the round stingray, the barb is toward the end of the tail. So if you're to strike the tail, that barb can impale some predator or something else, maybe a human or prey, etc. But if it's attached here at the base of the body, you can wave your tail, but you're not going to have any contact with the barb. So we're thinking that maybe this is some type of sexual display. Who knows? There's so many hypotheses that are open for figuring out. So if somebody in the classroom would like to run with that idea, we should chat. So anyway, back to the round stingray, another type of myliobatid. So here again, we have this stingray that is extremely abundant here in Southern California, but you find them across the entire coast. I'll get to that in a second. But one of the neat things of these benthic organisms, they feed on all types of snails and other benthic critters. But one of the weird things is how they actually reproduce. So all stingrays, all rays for that matter, are viviparous. They're live bears. But this particular species actually made the news about a year ago. You might be familiar with Charlotte the stingray. So this is a round ray that was in an aquarium in Hendersonville, North Carolina, over in the Appalachians, far from the coast. But she was thought to have become pregnant without ever being in the presence of a male round stingray. So maybe you're familiar with this. This made the news for a few months. And basically, there was 2 things that might have been responsible for her pregnancy. So the term parthenogenesis, that's basically where a female is able to give birth without the fertilization of any sperm. There's basically these weird little polar body cells that act as the sperm to quote unquote fertilize the egg. But again, there's no presence of a male. And since this particular female stingray was never in the presence of a male, maybe there was parthenogenesis. The other thing was maybe there's sperm storage. Maybe this particular ray was impregnated by a male before she even got to this aquarium. Who knows? But then some people were floating the idea that this particular shark actually impregnated the ray. And that's completely impossible because the male parts of this shark are not morphologically compatible to fit in the vent of this stingray. Plus, there's a bunch of genetic issues that completely mismatch this. So anyway, if you're unfamiliar with the male parts of a chondrichthyan fish, a cartilaginous fish, you're looking at the ventral surface, the bottom side of a shark, and what you see in this red circle are the claspers. So basically, it's the spermatic duct. So that'll be inserted into the vent or the cloaca of the female to get the sperm into the female. So this was completely not how that ray got pregnant. Maybe Saturday Night Live has the answer. So for those of you paying attention to certain cultural things, Charlotte the Stingray, a round stingray from California, made the news, which is pretty cool, I thought. But nevertheless, to be tongue-in-cheek, unfortunately, this particular stingray, there was no parthenogenesis, there was no impregnation by Michael Che, there was no shark involved, there was no sperm storage. It was actually a tumor, and the ray wasn't even pregnant. So anyway, that was sad, but at the same time, talking about parthenogenesis and all these weird sexual strategies for stingrays and other fishes made the news. So it got people talking about all these really interesting concepts that most people would never ever think about unless you're, basically interested in fish or just weird animal behaviors. Okay, so I digress. So back to just the geography of these fish. So in terms of where you find these round stingrays, the red line is basically representing where you find these particular species. So up in Humboldt, Arcata, Northern California, all the way down to Panama, but they congregate mostly in Southern California. So you can find these in Monterey Bay, but they definitely tend to occupy areas with warmer water, calmer habitat as well. So if you focus on this little yellow box, here we are in Southern California. So here we've got Long Beach in the north, La Jolla, San Diego in the south, Catalina, so the town of Avalon, about 20 miles off the coast. You find these rays very much abundant across this whole range. So I'm going to focus on this little area in this yellow box, and that takes us to Ray Bay. It's the local term for Seal Beach, which again is just a couple of miles from our campus. So we go out to Seal Beach to collect our rays, and this is what it looks like. So again, we want to figure out an answer to this question: what do these tail strike behaviors look like? Because we need to understand something about what these rays are doing to find a way to keep ourselves from getting injured at these beaches in Southern California. So here's what we did. So you're looking at some drone footage from a former grad student in the shark lab. So here's us doing a beach seine. So for those of you unfamiliar, you have this net that has floats on the top, weights on the bottom. You basically walk this out into the water. And we were doing this with the Orange County lifeguards. So we actually attached one end of the net to their jet ski, who helped take this out into deeper water. You basically make a half moon shape in the water, and there's poles on the end. So this army of my students, we basically pull this net back onto the beach, and presumably whatever's in the water column in that U-shaped area is brought onto the beach. So this is how we do it in Belmont Shore, Seal Beach, and other places along Southern California's coast. So just to show that in a little bit more detail, this red line is representing that U shape that we made with the beach seine. It's about 4 feet in height, and it was about 100 feet in total length. So that's allowing us to capture a lot of rays, or none, depending on that particular tow. So here is what it looks like when we get up onto the beach. And here's a video. We actually had ABC News covering us. Dr. Chris Lowe is in the red board shorts. I'm up at the top with that tan hat, and we're trying to figure out what we want to take. So we're basically selecting a mix of both male and female sexes of the round stingray and a size range. So we can do some ontogenetic studies throughout their life history to figure out if their behavior changes across between sex, and across sizes. So you'll also notice some people are taking buckets of water to keep them happy. So we are trying to process them as fast as possible, because remember, they stab really, really well. So we don't want them to impale their neighbors or themselves with their barb. So we quickly weigh them, measure them, let most of them go, and then take a few back to our campus. Okay, so bringing them back to the Marine Lab at Cal State Long Beach. This particular photo, we had 350 round stingrays in that one tow. Our record is about 880. Sometimes we get 0. So these are most common in nearshore areas in the summer and fall. But when you get into late fall and winter, they tend to go a little deeper offshore before they start returning to nearshore habitats in the springtime. So they're really just starting to come back toward the really shallow areas right about now. So we take a subset of those, we bring them back to the marine lab. We have a couple mesocosms where we have the small and medium-sized rays inside. We've got a larger mesocosm for the bigger rays as well. And here's one. So we feed them a mix of squid and shrimp 3 times a week. So they're quite satiated, very happy little rays. And I have my army of undergraduate students helping out with all of these things. So in terms of the methods for how we are going to evaluate the tail strike behavior, here's 2 of my former undergrads. I'd like to bring to your attention, you're looking at a tank. It's 180 gallons, filled about halfway. The seawater 16 degrees centigrade. You've got this one high-speed camera on the side, and we've got another high-speed camera on the top. So what we can do by having a minimum of 2 high-speed cameras, we can basically calibrate these cameras and recreate the 3-dimensional space. So when we step on these rays, by simulating that with this fake zombie foot. So if you see my former student Jacob holding this crazy cadaveric looking stump of a foot, we actually got that at Spirit Halloween store. So totally ridiculous. It's gone viral, which is super fun. I filled it with sand. I stuffed a PVC pipe in it, epoxied the whole thing, and we basically use that to simulate what it's like to step on a ray at the beach. So we record all of these videos, and here's what we do to calibrate that space. So what you're looking at is nothing more than a glorified test tube holder. So what we do is put these digital markers on the lateral view, and then digital markers in the dorsal view. We import those photos into MATLAB software. And by putting these digital marks in 2 different camera views, I can recreate the XYZ plane of this space. So when the animal actually does some motion in that space, I can put digital marks on that animal, and I can track the speed, the acceleration, all these other metrics of what it's doing behaviorally. Okay. So here is another student of mine, just basically representing what we do with the stingrays. And we have our pseudo-cadaveric force application device, the PC FAD. But in more fun terms, it's literally our zombie foot. And it just so happens to have this fake bloody stump, which looks really creepy in the videos, which I'm about to show you. So here you're going to see some tail strike behaviors. We film this at 500 frames per second. So just for reference, when you use your smartphone to film anything, that's at 60 frames per second. So this is a whole order of magnitude faster. And we basically will then import these videos into MATLAB, and we'll go frame by frame by frame to figure out what's going on. So if you want to see, if I can bring to your attention the video on the left, you'll notice that the left side, that's the snout, and then you'll see the tail, and then you can see the tail in the dorsal view. So we're going to step on the ray and pay attention to what's happening to the tail, and when the barb becomes exposed. Okay, so it's going to slowly approach the animal. It's going to make contact with the center of its body. The tail comes up, it strikes the fake foot, and you'll notice that the barb becomes exposed, generating this puncture wound and possibly releasing venom into that puncture wound. This whole behavior takes place in about a quarter of a second. So you snap your fingers and it's done. That's why we're filming this at 500 frames. So it's not the fastest behavior, but it's definitely fast enough. This is what it looks like when you step on a ray at the beach. Not fun. So here is a still image sequence of what we're doing when we import those videos into MATLAB. So if you notice the little red circle, that's representing the tip of the barb. If you see the little yellow diamond, that's representing where the barb is literally attached to the body. So in this still frame sequence, on the top left, that's time 0, the frame right before the behavior starts. And if you look at the bottom right, that's basically where the barb made contact with the foot. So again, that whole behavior takes about a quarter of a second and really, really, really fast. So again, we're able to put these digital marks and make sense of this in 3D. So here is our shotgun blast of data. So if Mike Graham is in the audience or Jim Harvey, they're going to hate me because I'm not really talking about a lot of stats at all today. But nevertheless, here's our stats. So on the x-axis, you've got the total length of the individual, so going from small to large. and we had 16 different individuals being represented. The y-axis is the raw velocity, meters per second. So on average, the animals generate about half a meter per second. But what does that mean? That's pretty much useless unless you put it into proper context. So when we scale this, with respect to the body length of the animal, the number of body lengths it generates per second in terms of how fast the tail is moving through water, it's about 2.7 body lengths per second. So just to put that in proper context, think of Michael Phelps, the fastest Olympian swimmer ever. So he swims at about 1 body length per second. So this is almost 3 times faster. But what does that actually mean in the animal world? So think of a scorpion. When a scorpion is striking its tail, it moves at about 2.5 body lengths per second. But keep in mind, a scorpion is doing this in air. Stingrays are doing this in water, and water is a very viscous, syrupy, dense fluid compared to air. So these animals are compensating for that in a really fast way. Notice there's no pattern whatsoever between the small, medium, and large size rays. So it's really a shotgun blast in terms of what we see. There is so much variability. Each vertical row are different trials of each individual. So again, you can see sometimes they're faster, sometimes they're slower. But if you just look at their maximum performance, they're basically moving at about 1.5 meters per second, or more or less about 3 body lengths per second. Okay, so what we do after we film all the rays, we take photographs of them and we make measurements of their body length, which is the snout all the way to the base of their body. We take their total length, which is their snout to the tip of the tail. We get their disc width, their left pectoral fin to the right pectoral fin, and the surface area of their body. So we can also start comparing how speed might change across different metrics of their body, because we're interested, does their behavior shift when you go from small to medium to large size, which is basically a proxy for age. And as you saw in the previous slide, there was no pattern whatsoever. There seemed to be a trend a little bit, but not really. There's just so much variability. So how do we do this? So because these are basically danger pancakes, they're a flat disc with a barb. Here's a former student basically using a spatula from the dollar store to help manipulate where this ray needs to go so you can keep your hands as far away from that barb as possible. So that's just one nice little way of repurposing a very simple kitchen device. So I can thank Moss Landing Marine Lab for helping make me a very scrappy scientist by just trying to be creative with not always having all the funds in the world. So still trying to find a way to do really good science. Okay, so what I want to do next is describe how round stingrays respond when you step on different parts of their body. So in the previous study, we always stepped on the dead center of their body. And the center of these animals is where they concentrate all of their main organs. So what happens if you step on their snout or their side fin or pretty much anywhere other than their center body space? So here is how we subdivided the body of the ray. So in this video that you see here, we're stepping on its pectoral fin and it actually just escapes. It tries to swim away. It's not actually generating a tail strike. So the way we subdivided this regionalization of the animal, S is for the snout, the nose, L is for the left pectoral fin, R is for the right pectoral fin, M is for their mid-body. Now, I want to bring to your attention, if you look at that weird little notch immediately to the left of the L, it looks, I call that my little pizza slice. So these animals, we have 18 of them in the lab. So to make sure we know who's who, because they're different sizes, different sexes, but they look very similar, other than the males and females, we make little pizza slices. I take a scalpel and I make a little 2 to 3 millimeter incision in different regions of their fin. And that will help me identify who's who so we can track them through time. Now that's living tissue. So that tissue grows back every 3 to 4 months. So I have to then re-notch that animal by remaking that spot, remaking that notch. So if you remember earlier today where I showed you a photo of my hand in that stingray tote bag, that's because I was re-notching the ray. My hand slipped, that was holding down the net, that was holding down the tail, and it struck my wrist. So anyway, that's the whole reason for that. Okay, so we've got these 4 different regions. what does this look like? So in terms of the results, again, here's the 4 different regions of the body: the snout, the left and right pectoral fins, and the mid-body. So you're looking at 4 different behaviors on the x-axis, and then just the raw number of times these animals actually did that particular behavior. So I want to bring to your attention that most of the time, these animals just escaped. It was very rare that they would ever generate a tail strike, except when you step on the middle part of the body. So these 2 red arrows are noting when you step on their mid part of the body, 85% of the time is when they generate a tail strike. So you can almost guarantee they're going to tail strike you, whether it's coming vertically, like you saw in the video, or it's coming from the side or any area in between. But most of the time, if you're not stepping on their mid-body, they just try to escape. And it's probably because they think they can get away. But if you step on their mid-body, you're literally pinning them down, and they're risking injury, because that's, again, where all their organs are, their main organs are concentrated. So it's more or less a life or death situation. So if you generate that tail strike, you're probably going to impale that predator, and they might release, and then you can swim away and survive. So again, 85% of the time, they're generating a tail strike at the mid-body. Very rare that you would ever find that anywhere else. Okay, so just to kind of sum up that study, or these 2 studies, the tail strike is reaching velocities up to 1.5 meters per second, so about 3 body lengths per second. The maximum strike velocity and force increased with the length of the stingray. I'm actually not showing all of the data, but we did find some significance there. But again, the main take home, when you step on the mid part of their body, that's when they're going to generate these tail strikes. And again, it's because you're pinning them down, and they're most likely trying to protect their internal organs. Okay. They escaped most often when you were stepping anywhere else, and it's probably, again, just because they have the opportunity to escape. There's a future study we would like to figure out how much energy is involved in escaping versus generating a tail strike, to see if there's any energetic component to that decision. Okay, so the next study I would like to briefly talk about, does the tail strike response have anything to do with the size of their feet? So again, if you have a big foot, a small foot, does the surface area affect how they respond? So we basically used these. So we 3D printed a bunch of feet that were actually the dimensions of my foot, some of the feet of my lab mates, and actually my nephew. We used the dimensions of his foot. And it's all basically just silicone, EcoFlex silicone material. This one over here we did not use because that's actually the size of a toddler, excuse me, an infant. And it's very rare and definitely not ecologically relevant that you would find an infant stepping on a stingray. Unless a parent is bobbing them into the water, I would call Child Protective Services. That would be a whole other can of worms. So we used a small foot, a medium foot, that's the original zombie foot, and then a large foot. So based on these 3 feet, we repeated the regionalization study to see how these might affect the stingray responses. So again, there's the 4 regions: snout, left and right pectoral fin, and the mid-body. But this time doing all of that with the small, medium, and large size fake feet. Okay, so again, not again, because you haven't seen it yet, but the trends were very similar to the previous study with stepping on their mid-body. So here we get a vertical strike, a horizontal strike, sometimes escapes. Where it says other or no response, sometimes the rays didn't respond, but the other is where maybe they were half committed. The tail started to go up or toward the side, but it didn't go all the way through. So we just categorize that as something other than a true strike. But nevertheless, when you step on their mid-body, the majority of the time, they would generate a tail strike. It was very rare they would do any other type of behavior when you make contact with the mid-body. So this was for the medium-sized foot. Here, if we're looking at the small foot, very similar trends. Again, with the with the strikes, that's happening at the mid-body and very much not happening anywhere else. But notice in the small foot for the vertical strike, there was not one time that the small foot elicited some type of vertical strike. And we're thinking that maybe because the small foot is so small, you could just do a little side strike, and you're still going to reach the foot or the ankle. So that was kind of interesting. We again want to revisit this idea of, is more energy needed to generate a vertical strike versus a side strike? So that's something we have yet to test. But either way, the same pattern held true, where when you step on the mid body, they generate a strike, whether it's from the side or vertical. So the size of the foot had some indication where maybe the surface area would influence the behavior, and in this case, not resulting in a vertical strike, but many side strikes, horizontal strikes. I'm not showing the data for the large foot, but it was the same exact pattern as the medium-sized foot. So just to kind of sum up that, oops, sorry about that. Again, where you step on them is absolutely crucial to the type of response you would expect. Okay, so just a quick aside, the way we're doing this, if you remember from the video and one of the photos, we're basically using our hands to press down on the body of these rays. So we need to know what the forces are that we're actually applying to the stingrays, because I'm doing this. One of my 15, 16 undergrads are doing this. So we might be applying different forces to the body. And that might be a major constraint and limitation on how you get a certain behavioral response. So what we did was visit a colleague's lab of mine here at Cal State Long Beach who does human biomechanics. So here's some of my students stepping on a force plate. So we're trying to figure out how much force do we generate with a regular footfall to simulate what that would be at the beach, but also we'll use our hands with the fake zombie feet to press upon this force plate to compare, is this close between all of the students, and is it relevant to what we would expect in the forces generated with a footfall? So just so you know, 100 newtons of force is basically 22.5 pounds of force. So I'm 150 pounds. So I should be generating about 650 newtons of force. So here, this is the pattern of my footfall. So we did 3 trials, so basically 3 footfalls per student plus myself, just to get an average of what forces we would expect with stepping on a force plate, just taking a regular step. So it's pressing down because, again, we're pressing on this force plate, and it's about 650 newtons of force that I generate. And again, that falls locks and step with what you would expect based on my body weight. So here's me. The video is not playing, sorry about that, but nevertheless, here's me pressing down on the force plate with the foot so I can actually compare that to my footfall. So when I'm doing this with all of the different zombie feet, here is the small zombie foot. That generates about 120-ish, 130 newtons of force. The medium-sized foot, also about 150-ish newtons of force. And the zombie foot, that's the large size, also about 150 to 200 newtons of force. So if you look at those values of force that I'm generating with my hands versus my footfall, it's only about 25 to 30% of what I'm generating with my actual step of my foot. So we thought, okay, this is a major problem. But then we realized, wait a second, we're doing this in air. When you're actually taking a step in the ocean, think of Archimedes' principle, and you're being buoyed up by ocean water, by salt water. So we found some papers that some scientists looked at the forces generated by people stepping in the water, and it's about 25 to 30% of what you would expect compared to your footfall on land. So we were right on par with what we expected, which was a huge sigh of relief, meaning that, okay, we don't have to throw out any of those data because it's actually ecologically relevant to the physics of the ocean and what we're doing in the lab. Okay. So the last study that I want to briefly talk about is there some type of surf booty material that can reduce these stingray-inflicted injuries? So here is Seal Beach. That's the pier. This is where we collect our rays. Here's a nice day to get some nice lefts. But again, you want to make sure you're not actually getting injured by a ray, because that's going to make it really problematic when you're trying to surf and swim and do all the things at the beach. So we are collaborating with 2 different groups who have made a very interesting composite material. So I want to tell you a little bit first about neoprene. So neoprene, as all of you know, at Moss Landing Marine Lab, whether you're scuba diving or you're surfing or you're doing whatever to not get cold from Monterey Bay's cold waters, you have this nice fabric material with a spongy foam. It takes about 1 to 2 newtons of force for causing something to go through that. So if you think of a needle, which is basically analogous to a stingray barb, anything sharp is going to go through almost any material. So again, it takes about 1 to 2-ish newtons of force to go through just regular neoprene. So that's a problem. That's not going to prevent you from getting stung by a stingray. But then the alternative is, well, if you're wearing chainmail or body armor, that's going to prevent a stingray barb from going through, but then it's really heavy and you're going to sink and you're going to drown. So that's no good. So what can we do to compromise this condition? So here is a material, this rubber composite with this polygonal shape that is basically manufactured to fit inside neoprene layers. So the group that we're working with, they made a single layer of this rubber composite sandwiched in between neoprene, and then a more rigid double layer also sandwiched within the neoprene. So we basically took the single and double layers and asked this question: what amount of force is going to cause this material to fail? So based on that original tail strike kinematic study, we calculated that the forces generated by the actual round stingrays are about 8 or 9 newtons of force on average. So here is us going back to the beach to collect some rays. We bring them, so when we're at the beach, we clip their barbs and then we release the animals back into the ocean. There's no pain receptors in this part of their body, so it's basically analogous to clipping your fingernail. So it's no problem whatsoever. So we take those barbs back to the lab, and here's what we did. So we have 8 different neoprene samples, 4 that were single layered, 4 that were double layered, and we would take 1 barb per neoprene sample, and we would stab each neoprene sample with that 1 barb up to 5 different times in different regions of that neoprene sample, because we needed to make sure that the sharpness of that barb doesn't become dull. So that's why we used 8 different barbs in this study. Okay, so here, just to be absolutely ecologically relevant, because we're not putting these neoprene samples against human skin, we have EcoFlex material, which is basically simulating the hardness of human skin. So we're using EcoFlex 30, which is basically the equivalent to your calf or your forearm. So not super flexible, but not super hard. So here's my student pressing upon that. So we would take the neoprene material, we would place it directly on top of this EcoFlex simulating human skin. And then here's what it looks like. And then we would go and use this machine. So here's a colleague of mine at Chapman University, and we're looking at a material tester, specifically the Instron material tester. And what I want to bring your attention to, this red box is a load cell. So we're able to manipulate the force generated by this load cell. And then here is a little clamp immediately below it. Attached to that clamp is the stingray barb. And we're moving at a pretty slow speed, 0.001 meters per second, and we're basically ramming the barb through the material. So the material is right over here, the barb is going to go through it, and we're literally recording the amount of force it takes to cause this material to fail. So here's what that barb looks like clamped, and here are the results when the barb goes through that material. Here you can see the tip of the barb sticking out on the left and the right. And here are the raw data. So on the x-axis is time. So you can see that the force, which is on the y-axis, it's ramping up as the barb makes contact with the material. What you see in blue, that's the double layer. What you see in the black hashed line is the single layer. So you can see it takes less force to cause the single layer to fail, as you would expect, because the double layer is much more rigid. So it takes about 12 newtons of force to cause the single layer to fail, and about 15 to 20 newtons of force to cause the double layer to fail. Now, one thing that's kind of cool, if you notice all these seesaw shapes toward the top right of this, that's actually just an artifact of the morphology of the barb. Remember, the lateral margins of the barb are serrated. So as the barb was literally puncturing through this material, the machine caught that, which is pretty cool. So anyway, just kind of fun where it's literally reflecting the biology of this particular structure. Okay, so here's just another way to sum up those results. So we have the double layer on the left, the single layer on the right, and we're looking at the load of force it took to cause these materials to fail. Just to remind you, this horizontal red dashed line, that's the amount of force that is generated by the round stingrays on average. So you can never guarantee 100% safety with this material, but you're going to be much better off with this material on your body to withstand and prevent a stingray from actually penetrating your skin. Okay, so to sum that up, the forces generated by the rays are less than the amount of the force to cause the material to fail for both the single and double layers. And as you increase a layer of material, it's going to increase the amount of force needed to cause that to fail. Okay, so here's some things that we're working on, and then I'll kind of wrap all of this up in a minute. Hopefully we'll have time for a Q&A. So what you're looking at on the left, this is a very flexible force sensor. It's basically a pressure sensor. So we would like to place that on the actual foot so we're getting in situ measurements of the forces generated. However, this material is about $10,000 and a sharp object will go right through it. So we'll basically get a trial of one. And I don't think it's financially worth it to do that. So there's other ways we're trying to figure out to get better in situ measurements versus just these back of the envelope calculations of what these animals are doing. So that's one little thing. Oops. Now you can see, there we go. Is it going to strike? It sort of does and goes away. Okay. So here's some things we're working on currently in the lab. You're looking at a nice diverse array of all of these stingray barbs that we've clipped from these animals. So one of the things we're using is Slicer. It's a type of 3D software, and we've micro CT scanned all of these images. So a micro CT, it's basically a three-dimensional X-ray scan. So we import those micro CT images into Slicer, and we can look at really high acuity, fine resolution details to understand the morphology of these barbs. So here it is. And then just to focus on one, what we can do is look at the curvature of the barb, the number of serrations, the way the serrations are accessing the long axis of the barb, the sharpness of the barb, the thickness of the barb, the curvature, all of these things. Now, one thing I briefly mentioned, these stingrays, they naturally shed their barbs in the spring and they regrow a new one by fall. So sometimes they have multiple barbs stacked up, but they do naturally shed these barbs in the spring and summer, really. So they're always removing them and replacing them. We've noticed that the same individual might have a different barb type. It might be straighter, it might be more curved, thicker, thinner, pointier, less pointy, all of these things. So there's so much variation within one individual. We're finally starting to quantify that since nobody's ever done it. And we're just trying to figure out how this looks within this species, but what it might look like across multiple stingray species. So to sum up the whole study, does the stingray shuffle actually work? So by shuffling your feet, will you stay safer at the beach? And the answer is yes. If you're shuffling your feet, what you're not doing is coming in contact with the dead center of the animal's body. And we already know that if you step on the center of their body, you're most likely going to get stung because they generate a tail strike. But when you contact their snout or their left or right pectoral fins, they tend to just swim away. So again, there's no 100% guarantee of safety. I've been stung by shuffling my feet, and it's not fun, but it definitely helps reduce the amount of injuries. So we put some science to this silly little statement in Southern California. So here's some things that we're doing and some other things. We just wrapped up a study looking at how these animals bury themselves. So we noticed that in between the trials of when we step on the rays, they bury themselves for camouflage, possibly to hide from a predator, or maybe they're hiding for prey so they can sneak up on them. we've noticed that they have a very coordinated behavior. They move their snout like a shovel, they undulate their side fins, and they oscillate their tail. So we're interested in looking at the neuromotor control of what generates these behaviors to move the sand right on top of their body. So if you look at the dorsal view on the right, notice that the sand doesn't really go chaotically into the water column. It's very finely controlled where it goes on top of the animal to bury itself. So we're looking at all of that. We're very much in the preliminary stages of analysis, but a very controlled behavior, which is pretty neat. And there's really not much known on flat fishes. So here's something that we're trying to expand upon. So another thing, we're collaborating with another group, Dragon Skin. You can actually go and buy their surf booties now, but what you see, two of my undergrads, we're using a 3D scanner to look at which parts of this surf booty is covered with this quote unquote stingray resistant material and what's exposed with just neoprene, so we can further prototype a better surf booty design for surfing, diving, etc. So here's just some other things we're doing. We're about to start a study looking at the cells that actually produce the toxins and how widespread or hyperlocalized that might be in the tail. And then looking at some electromyography. So trying to pinpoint which muscles are active or not in the behaviors to generate this tail strike. So I definitely did not do all of this by myself. I've got lots of undergraduate students who've helped me with all of these studies. I'd like to thank my funding sources. And as a shameless plug, if you'd like to follow us on Instagram, CSULB Stab Lab, definitely check us out and let me know if you have any questions. We've got a lot of fun things going on. So thank you so much, and I'll open up the audience for questions.
Nice, thanks, Ben. That talk was a lot of fun. We had we had it muted, but if it was not, you would have heard people laughing like all throughout the presentation.
Good.
All right, questions. Who has a question? Jonah, go for it.
Sure. I'm interested to know if you have any results for the large feet, because I know on the presentation you showed the small and medium, and not the large, and I'm curious because I have large feet myself, and so I'm just wondering what my chances would be with a brown stingray.
Yeah. So the large feet, the pattern looked very similar to the medium-sized foot. So again, if you're coming in contact with the mid-body, they're going to generate a strike 85+% of the time. But if your foot is really large and it's basically transcending their snout, their left and right pectoral fin, and their mid-body, they will most likely generate a tail strike. So it's really a surface area question. So if you have ridiculously big feet, sorry. So just shuffle your feet and try to reduce those chances of coming in contact with their center.
Thank you very much.
We need to get you some of these special booties.
Yeah.
Another question.
I had a question.
Yeah.
Great talk, thank you very much. I liked the idea that stepping on the mid-body is, they'll elicit a response to protect their internal organs. And I noticed that there was vertical strikes and horizontal strikes. Is there like a full 180 degree radius of the strike that they can hit if they're stepping on a portion away from the mid side of their body?
Yeah, that's a great question. So when they do a vertical strike, sometimes it goes 90 degrees, so all the way up. But sometimes it goes a full 180 and it will make contact with the top of the foot. So we haven't completed our quantification of where all the barbs make contact, but it's all over the place. It's the top of the foot, the side of the foot, very rarely above the ankle. But yes, they will generate their tail to do a full 180. We actually just saw that last Friday with one of our studies. So yeah, but one thing that's kind of interesting, I didn't really talk about it too much. We have no idea why they generate a vertical versus a horizontal strike. They definitely tend to do a vertical strike. It's much more common, but depending on how we orient the foot, and depending on how we orient the foot size of that foot, it's kind of all over the place. So we don't know ultimately what is happening. But yeah, they will do a full 180 quite frequently.
Wow, thank you.
Yeah.
Duncan.
Yeah, great talk. Thank you very much. So I was curious about both the penetration of the arbor neoprene and the strike response in terms of pressure. Because you showed these units in terms of force, and I was curious, does the pressure change significantly between the different feet? Does that cause different responses? And then have you looked at the pressure applied and how that correlates to punctures in the neoprene?
Yeah. So just in terms of the barb you're referring to?
Yeah.
Yeah. So we haven't looked at that, but a colleague of mine at UC Irvine, he has a PhD student who's looking at the pressure applied and the penetration depth of the barb. So they've simulated how deep the barb is going based on a certain speed of the strike. So based on the speed, that's going to apply a different force. And the angle at which the barb makes contact with the foot is also very influential. Is it a direct, perpendicular motion, or is there some oblique angle at which it's coming, which will be a reduced force and less pressure? So all of those things. But we didn't look at the pressure of the barb directly. There's a couple other labs who are doing that, and we're trying to figure it out because we have another study where we're trying to pinpoint exactly that.
Bruno.
Hopefully that answered your question.
Yeah, Duncan?
Yeah, I was still curious about whether or not the pressure of the foot stepping on the ray changes.
Yeah. So because there were 10 of us involved in that study of applying the foot to the animal, we never directly measured how much pressure we're applying, which is why we went and simulated that with the force plate study, just to see on average what forces are we generating with our hands to the fake feet onto the ray. So I can tell you that the rays will only ever generate a tail strike when we're being pretty forceful. And that's what we were trying to mimic when we were pressing on the force plate in that other lab. So we were applying about 150 newtons of force to this animal. If we applied anything else, we would squish it.
I wonder if you could attach like different amounts of weight on top of your little pole, right, to sort of give it slightly different amounts of pressure.
So a colleague of mine suggested that, and we actually have actuators that we can put on it so we can measure the speed and pressure simultaneously for much more precise results.
Yeah, cool. Bruno, you had your hand up.
Yeah, thanks for the talk, Ben. I noticed that like all of your interactions with the rays involved stepping on it from above. Do you have any data and information that from like approaching it like from a little bit further away and kind of dragging your feet, which is, I believe, what I interpret the stingray shuffle a little bit more as?
Yeah, I'm so glad you asked that because I left it completely off. Yeah, we did that. So we took our feet and we just approached them, toes first, ankle first, side of the foot, and they never struck it. They just didn't respond, or they just escaped by swimming in the opposite direction. So that's a great question. And we quantified it, and yeah, they never ever struck. Doesn't mean that won't happen in reality, because I've definitely been stung by shuffling my feet, but at least in captivity with those simulations, they never did it. One other thing to note, and it's a future study, everything we're doing, it's in perfectly clear water, and the water is never that nice the whole year. So one of the things we're going to be doing is filming them in infrared. So we're going to have everything done in the dark to remove the visual cues to see if that influences their behavior as well when a foot comes on top of them from any angle and from any part of their body. So yes, we did the actual stingray shuffle and didn't result in strikes. And then the plan is to also do this without the visual cues.
Thanks. Yeah. Boring result for science, but good result for the stingray shuffle.
Amanda has her hand up.
Hi, Ben. Thank you.
Hey.
That was Super, super interesting. Loved it. I walked in a couple minutes late, so apologies if I missed this part, but apart from human feet stepping on them, why might rays do this behavior? Why might they have a vertical and a horizontal strike? Like, who would they be striking?
Yeah, that's such a good question because they clearly did not evolve this for humans. I mean, they've existed for many, many millions of years before humans were ever on the docket. So, why would they do this? So, a former grad student with Chris Lowe, Elizabeth John, studied the diet of juvenile white sharks. And she would conduct beach seines up and down all of Southern California. And one of their main diet items are round stingrays. And if you look in the guts or the mouth or pretty much anywhere in a shark, especially white sharks, you find barbs stuck in their mouth, their buccal cavity. You find them stuck in their stomach, their esophagus, all of those things. So sharks are basically their main predator. So if you can stab them, you're now going to be stuck to the shark and they're going to swim away with you. Or if you can at least release your barb, you might have at least potentially deterred that shark from eating you. So it's mostly sharks as their predator.
That kind of gets at what I was thinking, like why, like what are the costs of doing this? Like why are they so hesitant to do it? And I was wondering if either like the neurotoxin production is costly or, yeah, are they trying to avoid like having to lose their spine or trying to avoid like getting, the spine stuck, right, in whatever they're, so they only do it when they really have to.
Yeah, which comes back to something we found. Again, they never really struck unless we were contacting the center of their body. It was very, very rare. But, if you're a shark that's just, perusing and digging through the upper part of sandy substrate, you're probably coming in contact with their pectoral fins, their snout, or their tail. So they might not strike. They might just swim away. But in the act of striking, sometimes the tip of the barb gets sloughed off, it breaks, it cracks. Sometimes the whole thing gets lodged in and they can actually release the barb depending on the season. But then again, because they naturally shed them anyway in spring and summer, by the time fall comes around, they have a fully formed new barb underneath. So there's very little time in terms of when they're considered vulnerable without a barb. But yeah, they could lose it, in the off season, and then they might be SOL. But again, if a shark really wants to eat them, they're going to find a way to do it. So.
Any other questions in the audience, or is there anyone online that wants to ask a question?
I think we're still in the chat.
Is there anything in the chat? Let's check the chat. Greg just says, "Welcome back to Moss Landing, Ben." That was when he joined the
Yeah, I saw that. Thank you.
Yeah, awesome.
One last question.
One last question from Mike Wood here.
What is it about Seal Beach that leads to the higher congregation of rays there relative to other areas in the region?
Yeah, very good question. So why Ray Bay? So it's protected, it's warm, it's calm, and for whatever reason, they just aggregate all the time. There's a lot of food. So the San Gabriel River mouth is just north. There's an estuary, the Bolsa Chica wetlands just south. So they do move quite a bit up and down the coast and sometimes all the way out to Catalina, but usually congregating within that area. So, and it's just easy access. So we seine in other places, but because of the bathymetry, it's shallowly sloped, it's sandy, there's lots of food, and again, it's warm, and not too many juvenile white sharks. So you, again, find rays across the entire coast of California, but in terms of SoCal, Ray Bay, some parts of Carpinteria, Santa Barbara, San Diego, if you go to Mission Bay and La Jolla, there's a ton of rays. So I don't actually know if they're any more abundant in those areas, maybe, but because this is so easy access for us, and because people are getting stabbed there all the time because it's a big surf spot. So just by having high human population densities of where people are getting injured, that's one of the spots. But they do congregate in multiple places. They aggregate in lots of these places. So that's, again, why we want to have better information from the lifeguards, from people who report these injuries. What does that look like? Is it really just focused on Ray Bay? And what are some of the limitations to that? So we have a better understanding of where else we need to be somewhat precautious of where you're going to get a stingray sting.
Yeah, we do a bunch of beach stain surveys for our surf zone MPA sampling in Central California, but we mostly see them in Stillwater Cove, which is also probably the warmest, calmest like area that you can get here, you know, during the summertime. So I think
and that makes sense. I mean, maybe Del Monte Beach, because that's calm and warmish. So
yeah, awesome.
Yeah.
All right, well, let's thank our speaker. All right, thanks so much, Ben. We really appreciate it. That was super fun.
Yeah, thank you so much. Glad you enjoyed it. It was good to be back. So
Yeah, right on.
yeah, thanks everybody.
Yeah, we'll see you next time.
You bet.